In this video tutorial, we are going to better understand the function of the nucleus, the structure, as well as some various question and answers under this particular topic, which you can best use it at your advantage for any exams, as well as to better your understanding and skills. So let's begin with some explanation on the function as well as the structure of the nucleus. Let's begin. Before we begin to better understand the structure as well as the function of the nucleus, first we have some question and answers which we are going to share at the end of this tutorial, and we are going to share between 1 to 5 and even more questions. And if you want to have access to even more, then you want to make sure to go ahead to see how you can subscribe to this channel, so that once we upload any new updates on this particular topic, you are going to be the first to be notified to watch and understand exactly what we have to share. First, let's begin with the description of the nucleus before moving to its function as well as some question and answers. Firstly, if we looked at the nucleus itself, it is located at inside of the nucleus. It is a dense, duct structure which is circular in shape, and of course, the reason why it is dense and dark, you remember I've used the key term dense and dark circular structure. These are the description of what we have to use when it comes to describing the nucleus itself. It is dense, and of course it is also a dark structure. The reason why it is dark in its complexity is due to the presence of the high concentration of RNA and DNA, which tend to stain itself dark and dense circular structure which is found inside of the nucleus. And the reason why it is dark and dense, it is because of the presence of the high concentration of RNA and DNA. Now, if you look at the nucleus itself, this nucleus is containing the nucleus. And inside of the nucleus, of course, it is the section where there is the synthesis of ribosomes. Now, we have talked about how this structure, the nucleus, is dark and dense. There is a section of it which, of course, at the center of it, it is not that too concentration in the darkness of its description. This area where it is not that too dark in the structure contain of what we call the RNA, which is the ribosoma RNA. This ribosoma RNA, at some point in time, tend to see how they can fold itself and cling to protein. Once it happens like this, it forms what we call the ribosoma RNA, which of course it is this protein that has clinched itself is responsible for the synthesis of ribosomes. So when the RNA at the center of the nucleus tends to combine with protein, it forms ribosomes. So in other words, the nucleus is also the powerhouse or an area where ribosomes are being synthesized. When the RNA combines itself with this particular protein to form the ribosomes, what happens is at some point they tend to move out of this area through the nuclear pore and place itself at the region of the cytoplasm. At this level, it tends to finish process of the synthesis of ribosomes, which is essential for producing protein itself. What we are trying to say is that sometimes the synthesis of ribosomes itself is not being completed at the level of the nucleus. The synthesis begins sometimes at the level of the nucleus when the RNA has combined itself with protein in order to synthesize ribosomes. What happens is at some point the unfinished synthesis of these ribosomes tend to move out of this area through the nuclear pore and place itself at the region of the cytoplasm, where of course the synthesis of the ribosomes is therefore completed. Now, let's look at the structural representation of the nucleus and explain some key features or what we need to take note of it. Let's look at it before we proceed. If you look here at this diagram, which has been represented here, this is the nucleus itself, or the particular structure that constitutes the nucleus. You can see that, just as we have described, that the nucleus is a dense circular structure which is dark in its complexion, located at the area of the nucleus. You can see this is the nucleolus. The nucleolus is here. It is dense and circular, dark in structure, and you can see it is right here inside of the nucleus. Here, it is inside of this area that the synthesis of protein takes place. Inside of the nucleus, at the same time, also have what we call the ribosomal RNA or the RNA that tends to bind itself to protein for ribosomes to be synthesized. In other words, the nucleolus is the powerhouse where ribosomes are being synthesized. And sometime at the center of this area, which of course is not very dark as compared to the other region, contain what we call the RNA. The RNA tend to bind itself with protein for ribosomes to be synthesized. And at some point, when it binds itself to that protein and the synthesis has not been completed, it moves out of this area through the nuclei pore. As you can see here, it moved out of the nuclei pore and found itself at the level of the cytoplasm, which is around this area. Now, the nucleolus is being surrounded by some structures, such as the nuclear envelope, which you can see, the double membrane barrier that tends to cycle the nucleus, as you can see. It is called the nuclear envelope. And the nuclear pore also is a passage where those ribosomes, which need to be synthesized inside here, when they combine with protein, will move out of the system or out of this area through the nuclear pore to locate itself at the region of the cytoplasm where the ribosome synthesis will be completed. Also, inside of this structure is what we have what is called the chromatin. This chromatin is made up of heterochromatin and euchromatin. That is euchromatin and heterochromatin that makes or constitute what is called the chromatin. Now, of course, we have seen the structure of the nucleolus inside of the nucleus. And you can see that based on these, it is being circled by some structures such as the nuclear pore, the nuclear envelope. We also have chromatin found inside at the level of surrounding the nucleolus, the chromatin, heterochromatin, which make the chromatin itself. That is heterochromatin and euchromatin that makes the chromatin. Now, this being explained, we now understand or better understand the nucleolus, the structure and function. Now, let's look at some of the specific questions you need to know when it comes to this particular aspect. Let's look at some review of some questions as well as trying to answer some of questions which can be presented under this topic. Let's proceed with the first question. With this presentation, we have a total of 15 questions under this particular topic, but of course, we are going to be sharing some few, and in the next upload, we are going to see how we can share even more. So you want to make sure to go ahead to see how you can subscribe so that anytime we publish another series of questions with the answers, you are going to get it immediately, and you want to make sure that you enable the bell notification icon. Looking at the first question here, the first question states, the nucleolus is a darkly stained structure within the nucleus, but have you ever wondered why it appears so intensely colored under a microscope? Understanding this helps explain its critical role in cell function. Now they are asking the question, what exactly causes the nucleolus to stain so darkly compared to other nuclear regions? We have some specific answers here. The first option, it is full of cytoplasmic protein. That is, these are some of the responses. Response number A, it is full of cytoplasmic protein. B, it contains large amount of DNA and RNA. C, it has a thick nuclear membrane. And D, it contains a dense lipid and enzyme. Which of these are correct? The correct answer is the B, which contains the large amount of DNA and RNA. I have earlier explained that the reason why it is dense and so dark in its color is because it's stained with the dyes which is being used under the microscope. So it is very rich in RNA and DNA. That's the reason why when it is used under an electronic microscope to determine if that area is the nucleolus, it is because what happens is it tends to stain with the dyes which is being used. And once it encounters this dye, what happens is it stains so dark. This tells us that it is very rich in RNA and DNA. It is this that gives the feature of a darkness when stained with the dyes. This is why B is the correct response of this particular question. Let's look at question number 2. When we look inside the nucleus, we might notice one or more nucleoli. But how many nucleoli can typically be found inside a single nucleus? And what determines their number? Choose the most accurate reason from the option below. A. Only one appear in all human cells. B. It depends on the number of chromosomes. C. One or more may be present. D. It varies with the cytoplasm or the cytoplasm content. So the answer here is C. One or more may be present. So inside the nucleus, we can have one or more nucleoli. Question number 3 is, have you ever wondered what makes the nucleus a key player in protein production? It is not just a structure, it is a manufacturing hub. What is the primary function of the nucleus in the life of a cell? A. Transporting genetic materials. B. Synthesizing DNA for replication. C. Making ribosomes. D. Packaging chromosomes for division. The correct answer here is C. Making of ribosomes. So the primary function of the nucleus is for the synthesis of ribosomes. Remember earlier I've stated that inside of the nucleus we have what is called the rRNA that tends to bind with protein to synthesize ribosomes, which is the major key player in this rule. So it is at the level of the nucleus where ribosomes are synthesized by combining protein and rRNA to form the ribosomes. Sometimes it moves out of the cell through the nuclear pore to find itself at the level of the cytoplasm to continue the production of protein at this level whenever it is needed. So ribosomes, or the key function of the nucleus, is for the synthesis of ribosomes. Question number 4. The dense core of the nucleus is fascinating in structure and function. Do you know what is mainly composed of and how that contributes to ribosome production? Select the best explanation below. A. Only lipid-rich membrane. B. Random nuclei protein. C. DNA from one or several chromosomes. D. Mitochondrial fragment. The correct answer is C. DNA from one or several chromosomes. You remember I first of all stated that this nucleus is dense. That is, it is dense and compacted. That is dense, circular, and dark. That is a dense and circular structure. And I stated that the denseness of this nucleus, as well as the reason why it will stain with the dyes to give the deep dark complexion, is because of the richness of DNA and RNA. So the reason is C. Question number 5. Cell undergo nuclei division, and during that time, certain structure change significantly. What happened to the nucleus during nuclei division, and why might it appear to disappear under a microscope? A. It shrinks due to water loss. B. It disappears as DNA uncoils. C. It dissolves in the cytoplasm. And D. It fuses with the endoplasmic reticulum. So I would like you to go ahead to state the answer of these so that we are going to correct you in the comment section of this video. We want you to use your own thought and see how you can answer question 5 and give your response below in the comment section. We are going to end here, and we are going to see how we upload another episode containing more questions under this topic. So see you again in the next upload, and bye-bye for now.